We're now ready to move into Chapter 15 in our microbiology class. In this chapter, we are going to discuss what microbes do to become pathogenic. Different characteristics that microbes can have that can increase their ability to be a pathogen or sometimes even decrease their ability to be a pathogen. Before we begin, let's remember what a pathogen is. A pathogen is any microbe that has the ability to cause disease. In order for a microbe to become a pathogen, it must meet all four of the requirements listed here. First, a pathogen must gain access to the host. This means that a pathogen must have access to some portal of entry that allows that pathogen to actually go into the host in the correct area that that pathogen needs to infect. For example, if we think about a cold virus, the cold virus has to go into the body through the respiratory portals of entry, meaning that pathogen has to have access into your nasal or oral mucosa. That flu virus cannot hurt you if it is, for example, just sitting on your arm. It has to have the ability to access the host in the correct area. The second requirement, after the microbe gains access to the host, the microbe must be able to adhere or stick to those specific host tissues. Going back to our same example with the flu virus or the cold virus, that virus affects our respiratory system. So not only does it have to have the ability to get into our respiratory system, but once we inhale that virus, it has to be able to adhere, attach, stick to some portion of our respiratory system. If that pathogen cannot adhere, we would simply be able to exhale the virus and it would not make us sick. The third requirement for pathogenicity is that the microbe has the ability to either penetrate into specific host tissues or once it gets to the point of adhering to host tissues, the pathogen must have the ability to evade or trick our host defenses somehow. Now, this is going to be a little bit harder for us to focus on in this chapter because we've yet to go through our host defenses. That's where we'll be heading into chapters 16 and 17 talking about the immune system. So for now, we just have to accept that once the pathogen has access to our body, it is stuck in the appropriate area. It has to be able to remain in that area long enough to complete the fourth requirement, which is the pathogen must be able to directly damage the host. Remember, that's the definition of a disease. The pathogen has to cause some sort of change in the normal activity or function of the host tissues that the pathogen is living. There are some exceptions to this requirement. Some pathogens may not damage the host tissue directly. Instead, you may see a pathogen that causes that has some sort of normal metabolic process that leads to an accumulation of waste products in the host. And then indirectly, those waste products will then cause the disease state, the actual damage to the host tissue. Now if we look a little closer at each of these requirements of pathogenicity, let's first look at the requirement that the pathogen must be able to gain access to the host. The method in which a pathogen gains access into the host is through one of the so-called portals of entry. There are three common portals of entry: mucous membranes, skin, and what we call a parenteral route. The mucous membranes are by far the most common portal of entry, simply due to the amount of mucous membranes you have on your body and the biological and chemical makeup of those mucous membranes. You have mucous membranes anywhere on your body where there's an opening to the inside of your body. All of the linings of your respiratory tract, your gastrointestinal tract, your genitourinary tract, and portions of your eye are covered in mucous membranes. So all of these areas are considered part of this one portal of entry. This means we could give examples like the influenza virus, which you breathe in, that uses a mucous membrane portal of entry. If you eat some bad food and you get some sort of food poisoning, that would be the gastrointestinal tract. If a disease is spread sexually, then it is spread in one of the genitourinary mucous membranes. The most common of all of these mucous membranes is going to be the lining of your respiratory tract, simply because there are times when we may not be eating or doing some other activity, but we are always going to be breathing. So our respiratory tract is always exposed to pathogens. The second portal of entry, the skin, is actually much tougher than you might immediately think it is for a pathogen to gain access. Your skin is covered with keratinized, dead, waterproofing epithelial cells. So you actually almost need a crack or some sort of cut in that skin for the pathogen to gain access. Once you have broken the skin membrane somehow, then we consider the portal of entry not through the skin, but by a parenteral route. So something like West Nile virus that's spread by a mosquito bite, or you cut your hand with a knife and you get a staph infection, or you get a splinter in your foot and you get an infection, or you step on a nail and you get tetanus. All of those are considered parenteral because, yes, the pathogen did go through the skin, but the pathogen did not go through the skin on its own. The pathogen only made it through the skin because there was some sort of break in the skin, the bite, the cut, the splitting of the skin, something of that nature. If you recall back in Chapter 7, I made a comment on how I'm not a big fan of the alcohol-based hand sanitizers. And this parenteral portal of entry brings us back to that topic. Simply drying and cracking of your skin exposes your body to many more pathogens entering this parenteral route than if your hands were nice and moisturized. So just an extra little tidbit going back to our Chapter 7 discussions. Okay. So that was our requirement number 1, gaining access to the host. Requirement number 2 was that the pathogen has the ability to adhere or stick to the host. This picture right here shows you the pathogen in brown, and it demonstrates how, regardless of which pathogen we're talking about, the pathogen is going to have some sort of proteins sticking off of its surface. These are called adhesins. The adhesins are going to be specific for whichever host tissue that pathogen wants to invade. So if this was our flu virus, then these are specific receptors in our respiratory system on our mucous membranes. Once this pathogen has access to the area, these adhesins are going to allow the pathogen to stick to the appropriate cells. Sometimes the adhesins are part of a glycocalyx, sometimes they're part of flagella, fimbriae, or they can just be basic proteins sticking out of the peptidoglycan of a bacteria. These can be spikes on the envelope of a virus, anything like that. They are simply proteins sticking out of the pathogen that allow it access to stick to its host cell. We haven't talked much about biofilms in this class, so I want to throw that in while we're in the area talking about how pathogens can adhere. Biofilms are collections or little communities of microbes that all work together to stick to some specific surface. The most common place that we see biofilms are in our mouth. That's actually what this picture is here. This is an electron micrograph of a tooth. And all of the little purple circles, these are coccus-shaped bacteria that form dental plaques in your mouth. When you wake up in the morning, you can scrape the front of your teeth and you can get a white sticky substance off of your teeth. That's the biofilm. And the reason this fits into our discussion now, all of the bacteria in your mouth have adhesins that can specifically stick to the surface of your teeth, your gums, or the epithelial layers on the inside of your oral cavity. One way or another, the first pathogen has adhesins that are specific for that region. then the next pathogen may not be specifically able to stick to your cell, the host cell, but instead it may be able to stick to the pathogen that is already stuck to your host cell. So we just end up seeing large clumps of bacteria in certain areas. Now that our pathogen has gained access and it has adhered to the host cell, now we need to think about what can the pathogen do to cause damage or penetrate into our host defenses. And so let's first look at how the pathogen can kind of trick our immune system. By far the easiest way a pathogen can trick our immune system is simply having a capsule. And we'll go over this more when we get into Chapter 16, but what happens is when the pathogen has a capsule, which remember is the tough, highly organized type of glycocalyx on the outside, that keeps our immune system cells from being able to ingest and destroy that pathogen. So simply having the capsule allows the pathogen to penetrate or trick our immune system. A second example would be a pathogen that has a specific component in its cell wall known as the M protein. M proteins are resistant to heat and acid. Heat and acid are two ways that our immune system works to help kill certain pathogens that enter our body. One of the human body's responses to invading pathogens is fever. A fever, unless you're very, very young, is actually a good thing. A fever is your body heating up, hopefully enough to kill any pathogen that has invaded your body. Acid is present in specific areas in your body, most specifically in your stomach. That way, most things you eat, once they make it into your stomach acid, the acid is going to kill the pathogen so that it can't cause further damage once it moves further into your gastrointestinal system. Bacteria that have things called M proteins, they are going to be able to live in the presence of fever or stomach acid. So that allows them to trick our immune system. Some pathogens can produce enzymes that allow them to trick different parts of our body. And I would never expect you to know these in huge detail. Let me just go over a couple of them with you. One good example is the bacteria Staphylococci. Different species of Staphylococci can produce different enzymes. But one particular Staphylococci produces an enzyme called a kinase. The kinase dissolves blood clots. One thing our body does as part of the inflammatory response in a region is to form a blood clot so that any pathogen that has entered that area cannot leave. It's sequestered to one area. So Staphylococci has developed a way to produce an enzyme that can trick us into letting it out of the sequestered area where our immune system is working. Another example, Clostridium perfringens produces an enzyme called collagenase. Collagen is a protein component, fiber component found in the deeper dermal layers of your skin. Down inside this area of your skin, most pathogens can't make it through to get to blood, which is ultimately what the pathogen wants. Once it accesses your blood, it can move through your entire body. But Clostridium perfringens has figured out, well, if I can make this enzyme, I can break down the collagen, breaking down the tissues of the body, and that way I can move wherever I want. Now let's move into how pathogens not just trick our immune system, but actually damage our host cells. There are many, many ways that we could go over, but I just want to point out some of the most important ones to you. One thing that a pathogen can do is use all of our nutrients. And a great example of that would be some of the bacteria in our body that have a requirement for iron themselves. So they will actually start using up the iron that's in our blood. And many of you may have already thought about the fact that we need iron in our blood. It's in our hemoglobin. That's how we carry oxygen around. So that's a pretty damaging effect that a pathogen could have, stealing our iron. Something probably more obvious and easy to understand, if you have a tapeworm living in your intestines, everything you eat, he is eating too. So a lot of times people will see signs of malnutrition, weight loss, things like that, because the pathogen is using your nutrition instead of allowing you to use it. Some pathogens have the ability to directly damage us. There are crazy pathogens such as a brain-eating amoeba, and this is going to be a little protozoan that actually lives in your brain and destroys your brain tissue. In Chapter 13, we just went over prions. We know prions directly damage your brain cells and things like that. What I really want to spend time talking about in this chapter is how some pathogens can damage your host cells by producing toxins. There are two types of toxins that bacteria can make. They can make exotoxins or endotoxins. Typically, the exotoxins are the really, really bad guys. Exotoxins are things like neurotoxins that destroy your nerves, hepatotoxins which destroy your liver cells, cardiotoxins destroy your heart cells. These can be extremely serious. Endotoxins are still not any fun, but they are usually more of a kind of, I like to think of it as a revenge of the bacteria. Endotoxins are usually going to be things like a basic food poisoning type symptom that you may not be happy you have it, but it's going to go away fairly quickly. So let's look a little bit at how these two things differ. Here's an example of how the exotoxin is produced. And here's how I remember it. Exo sounds like exit. An exotoxin is made by a living bacteria that while the bacteria is still living, he makes the exotoxin and it exits the body of the living bacterial cell. So here's our bacteria. The bacteria makes the exotoxin. The exotoxin exits. This cell is still alive. The exotoxin can now bind to a host cell. The host cell will pull in the exotoxin by a process of endocytosis, simply pulls it in. The exotoxin breaks apart. One part of the exotoxin stays in the cell and damages the cell. If this was, for example, a neurotoxin, then once the neurotoxin was released, it would bind a nerve cell, and then this portion of the neurotoxin would stay inside of the nerve and eventually kill that neuron, that nervous system cell. Exotoxins can be extremely serious. They can cause all sorts of problems, from your basic type of nausea, vomiting, all the way up to shock, death, paralysis. The list can go on and on. This slide just gives you a few examples of the different things that an exotoxin can do. They can be extremely serious. If we compare an endotoxin to that of an exotoxin, the endotoxin is actually inside of the bacteria. It is part of the bacteria's cell wall. We only see this in gram-negative bacteria. Since the endotoxin is actually inside of the bacteria's cell wall, this bacteria has to die before the endotoxin is released. So this is what I mean by, I think of endotoxins as the revenge of the bacteria. So here's this endotoxin. We got it in our body, and now our immune system cell, this macrophage, he's going to kill that bacteria because he's not supposed to be in our body. Once we kill the bacteria, that then releases the endotoxin into our body. Because of this endotoxin, our immune system starts responding, and we get things like a fever, chills, weakness, and in some severe cases, endotoxins can lead to some anaphylactic shock, death, and we've also seen miscarriage in these sorts of infections. So I made this little summary slide for you so that hopefully you'll be able to keep these two things separate and straight in your minds. Let me go over this slide with you very quickly. The exotoxin, you can see here in the picture, you have a living cell still alive making exotoxin. Most of your exotoxin producing cells are gram-positive, and most of the time the exotoxin itself is simply a protein so it does not cause a fever-based response. The endotoxin, you can see here in the picture, here's the live bacteria. The endotoxin itself is not released until the cell dies. All of your endotoxin-producing bacteria are gram-negative. The endotoxin itself is composed of lipids or fats, so it immediately penetrates into different cellular components in the body, so you end up with always getting a fever response from the endotoxin. Now, this is not necessarily a requirement for pathogenicity, but we need to think about, in order for a pathogen to truly exist, it has to have a portal of exit. It has to be able to leave one host so it can go to another. So this slide just simply lists the different portals of exit that a pathogen may use. Pathogens may exit your body through your respiratory tract. This can happen by coughing or sneezing, or even just speaking, breathing. You can allow a pathogen to leave your body. Pathogens can leave out of your gastrointestinal tract through your feces or your saliva. They can leave by your genitourinary tract through urine or vaginal secretions, and also penile secretions, although I forgot to put it on your slide. Pathogens can exit out of your skin. However, again, out of the skin is really just as tough as directly through the skin by itself. There is one worm that we will go over when we go through diseases that actually pushes its way out of the skin when it's ready to leave the body. The last portal of exit is through the blood. So you could have a cut, a needle, some sort of biting bug like a mosquito can allow the pathogen to exit out of your body that way. To summarize what's important out of Chapter 15, you need to make sure you know examples of each of the 4 requirements of pathogenicity. And most importantly, you need to make sure you understand the difference between an endotoxin and an exotoxin.